All right. Good morning, everyone, and thank you for attending today's TED-style session on advancing genomics engagement. My name is Rowsey, and I am the chair of your program committee this year, and I have no relevant financial disclosures. And that's all my slides, but as you are all aware, the field of genomics is rapidly expanding, increasing in both scope and complexity. So due to this exponential increase in need, we have a need to increase the engagement that we're getting in genomics to keep up with that demand. It is that need that has inspired the selection of the speakers today. I'm happy to introduce all three of our speakers who will be sharing different perspectives on how we can continue in advancing engagement in genomics. First, Dr. Shoumita Dasgupta will be from Boston University, will be presenting on her experience and perspectives on mentorship and genomic education. By engaging across communities and by interacting with our learners authentically, we can set a foundation for a future of genomic medicine that benefits all patients. Next, Dr. Rebecca Ahrens-Nicklas from the Children's Hospital of Philadelphia will discuss her experiences on the first ever personalized gene editing therapeutics. By highlighting the potential of genomic therapies, she will showcase how we can have a multidisciplinary team focused to the singular goal of advancing therapies for our patients and how we can use this as a framework for future genomic therapies. And finally, Sharon from the Genetic Alliance will be presenting her journey into genetics with an emphasis on how the layperson can connect with genetic information. She will be highlighting the importance of connecting that genetic information to the people who need it most, which are our patients and their families, and then illustrating how patients and their families can use that information to advocate specifically for their needs, with an emphasis on the importance of collaboration. So with that, I'm happy to turn it over to our first speaker, Dr. Shoumita Dasgupta.
Good morning, everyone, and thank you to Ross and the program committee. It's a special honor to be here with you this morning, especially talking about mentoring in a room full of mentees, peer mentors, and mentors. I have no relevant disclosures. Does this image take you back to a moment where you felt like a fish out of water? Maybe you were the new person at school. Maybe you had recently immigrated to the country. Perhaps you were starting a new job or a program. This feeling of being a fish out of water can be incredibly overwhelming, and I often think about the bravery it took for my parents when they first came to the United States as immigrants in the 70s from India. They left behind everything and everyone they knew and loved in order to pursue the American dream. Like so many immigrants before them, they had to start over in many ways. Although they had advanced scientific credentials, in my father's case, his credentials in chemistry didn't transfer to the U.S. system, so he had to start over. And he eventually earned his PhD in polymer chemistry at age 39, which you can see in the photo on the right. That's me with him on graduation day. My mom actually was trained in psychology, and when she came to the United States, she started as a teacher's aide in the public schools of Ohio and reinvented herself in so many different careers in so many different ways over the years. The way that they were able to succeed and, really navigate all of these foreign and unusual systems and structures that they were unaccustomed to was partly through grit and tenacity, for sure, but also through the incredible benefits of having mentors and friends being willing to extend a helping hand. And this is something that I understand just implicitly through my own experience and my family's story. When my parents came to the United States, they had to move way outside of their comfort zone in order to head into the growth zone. And this is something that we as mentors can help people explore. We have been through this and we have experienced those bumps along the road, so we can help smooth this journey for our mentees. In order to help you understand some of my philosophy around mentoring, I want to share a little bit about my own journey. So I told you that my family story in the United States started in Ohio, but most of my formative years were spent in Pennsylvania, where we moved after my father graduated. In Pennsylvania, I was settled in central Pennsylvania, which is a primarily white area of central Pennsylvania. So I had to become accustomed to the idea of code switching between a primarily white school environment and then my chosen family of Indian immigrants. This was a real complex negotiation that I had in terms of navigating my environment. And when it was time for me to pursue higher education, I went on to MIT and I moved to Cambridge, Massachusetts. So this is a completely different environment where I was surrounded by people from all over the world who were asking those big scientific questions and pursuing paradigm shifting science. This experience was absolutely electrifying. And it wouldn't have been possible without the benefits of Pell Grants and work study, which really helped me make that transition from small town Pennsylvania to Cambridge, Massachusetts. Those Pell Grants and work study opened doors for me, and as soon as I arrived on campus, I started pursuing opportunities to look for that work study program. My very first lab job was actually as part of the Human Genome Project. I had a very entry-level job. I was washing dishes and pouring sequencing gels. But you know what? There was something incredible about being in that room with everybody pursuing this historic international effort. I was so excited about the science I was able to contribute to in my own small way. And my advisor, Dr. David Hausman, saw this excitement and thought, there's something in her. There's something that wants to pursue science. And he opened an opportunity for me to join his lab and pursue science through my own project. Now, this was really special for me because it allowed me to explore science and cancer genetics, and I stayed in that lab for the entirety of my undergraduate education. One of the most meaningful classes I took while I was at MIT as well was his class, actually, which spanned the junction of medical and molecular genetics. Sound familiar? And in this class, he specifically brought in patients to share their life stories to help us understand the impact of genetic variants on real life decisions and lived experience. David has always been a generous mentor, centering the patients, how our science affects them, and the learners. And he's been an important example to me to this day. Something that I've learned from him and his mentoring style really is to be able to see something in a mentee, perhaps even before they can see it in themselves, and open doors to make some of those opportunities possible. This can be life-changing. Also, while I was in college, I was able to pursue teaching opportunities. Some of the teaching opportunities I engaged in were part of the Experimental Studies Group, which is an alternative version of the introductory curriculum for first-year students. This alternative version had much smaller class sizes and was meant to ease the transition for students who might come from minoritized backgrounds or under-resourced schools. So this particular experience allowed me to take ownership of the introductory biology class for this small cohort of students and gave me a lot of autonomy and the ability to explore this interest of mine. I was also able to TA for our traditional curriculum biochemistry course. And these experiences really affirmed for me how excited I was about mentoring and really that teaching and mentoring go hand in hand. When it was time for me to move on to graduate school, I decided I wanted to pursue graduate school blending these interests, both at the lab bench and in the classroom. And so I moved out west to UCSF in San Francisco. While in graduate school, I continued to pursue teaching activities. One of these teaching activities was working with a teaching fellowship that taught professional students at UCSF. The professional students, the medical students, as well as the dental students, were an incredible training ground and really offered me insight into what a career in teaching could look like through my near-peer mentor, Dr. Tracy Fulton. Tracy was part of the same graduate program in me and showed me what it was like to be able to pursue a career in education specifically. I also had the incredible privilege to be able to teach in the California community college system. The community colleges in California are just such an incredible experience and such an incredible resource to make higher education accessible to so many learners who might not otherwise be able to experience it. I learned so much in that training ground. I was able to teach a class in human biology that met from 6 to 9 p.m. on a weekday. And you can imagine that my adult learners had already been at work all day long. They had rushed through dinner and come into class. And I had to hold their attention for 3 hours at this time of day. Not only that, but they were not STEM students. They were taking this class to fulfill a requirement. So I learned so much as an educator, thinking about how to make the message correct, meet them where they are, make it relevant to their own interests, and think about all the different factors that influence their educational journeys as well, beyond the classroom. This was a really important experience for me and has really shaped my ideas about what teaching and mentoring look like. This would only have been possible, and it was only possible because my mentor in the lab environment, Dr. Regis Kelly, when I told him that I was interested in teaching, He was delighted. He just was so excited that I found a professional space of belonging, and he encouraged me to pursue those opportunities rather than pigeonholing me into lab-only kind of activities. So I really think this is another aspect of mentoring that's really important, is thinking about having a whole mentoring team where each person brings different things to the table. Some people are there to help you with really direct, salient advice, but other people are there to help you expand your sandbox and explore more activities. When I finished my graduate education, I came back to the East Coast and I moved to Boston University, where I started a faculty position in the Department of Genetics and Genomics at that time. I was the founding director of the graduate program in Genetics and Genomics, and I also direct the medical genetics curriculum and racism in medicine. This was in 2003 when the Human Genome Project had just released its draft sequence. And when I arrived, I was hired as a full-time educator because that was my interest. That was both liberating and terrifying because there was not a clear path of what a full-time educator actually looked like. And so I was constantly second-guessing myself. Of course I was. Look how young I was at that moment. So in 2003, I started my journey on this path as a full-time educator, and I constantly felt like this. I felt this internal voice in my head that was really, second-guessing and asking, am I really here? Do I belong here? But while trying to present this calm, cool, collected exterior, the imposter syndrome was so real. But over time, I was able to start feeling more comfortable in my own skin and to start thinking about ways to bring my own lived experiences to the table. That has allowed me to grow and be more authentic in my professional activities and to really inhabit this space at the intersection of my core beliefs. That is a love of science, an awareness of equity and access issues, and an appreciation for the transformative power of education. And these continue to be my guiding stars. So because of my journey and because of my family's journey, I'm quite aware of all of the different bumps on the road that can happen as you're navigating these different transitions. So I think as mentors, we really have the ability to smooth those bumps by opening doors and helping our students connect with different activities that they might not even realize they have in them. So in my own mentoring style, I really think it's important to ask very intentionally, in what spheres do I have privilege, and how can I leverage that privilege in service of people who don't have access to the same privileges? I think this is really important because, in fact, our messages are amplified. They go well beyond our professional roles in classrooms, clinics, and laboratories. And so we have to be thinking about how we can navigate this on behalf of a larger constituency. This was part of what drove our work in the context of a safety net hospital, where we realized, what our authority levels were, not only just within our own institutions, but also that our words mattered to the general public. And so at Boston University, which is a social justice medical school, we started engaging in curricular-led reform. And this project was really spearheaded by a group of students who wanted to examine the strengths and weaknesses discussing race, racism, and medicine in our medical school curriculum. This advocacy work was an incredible privilege to be a part of, and we actually completed our self-study and came up with recommendations just in the summer of 2020, which, if you'll remember, of course, was the summer that George Floyd died and the Black Lives Matter movement was reinvigorated. At that moment in time, our community was very hungry for advice about what they could do to try to promote equitable change in medicine. And so we were really incredibly honored and humbled to be able to share some advice to give our community some action items in that regard. And our process and our findings were published in this paper. That experience really helped me understand in a very visceral sense how important it is to be able to talk about the intersection of identity and science. And science tells us a lot about different aspects of identity, but there is a limit to what science can unearth, and identity is a very separate, socially defined process. The conversations I was able to have with my students over the years were incredibly meaningful to me, and I hope they were as meaningful to them as well. I wanted to be able to extend these conversations into other spaces as well. And so it was very important to me to be able to bring these lessons together in the form of a book, Where Biology Ends and Bias Begins, to try to help people have these conversations in their classrooms, in their clinics, but also in their libraries, in their museums, at their dinner tables. I think this is something that we can really center intentionally as mentors, is how do we create these safe spaces? How do we make a space that our mentees can actually see themselves in their profession, in their science, and in their mentors. But if I'm being honest, we in genetics still have some work to do in this regard. If we look at this graph in orange, you'll see the fractions of American populations in different racially defined categories. And in blue, we see the fractions involved in the genetics workforce. So you can see that there are a number of underrepresented groups just in this racially defined category. But of course, this is true in many other social identities as well. Now, this is a problem because diversity in our teams really matters, because having diverse teams helps us address healthcare disparities, helps us be more productive as teams, and helps us to deliver culturally competent and culturally informed care. The easy answer about why this is the case is to blame it on the pipeline, which for sure is part of the issue, but that can't be the whole story, because there are 5 to 10-fold fewer BIPOC geneticists than there are other specialties of medicine. So it's not just the pipeline. If we look at the geography of where our geneticists are spread in the United States, you'll see a familiar pattern where we have concentration of geneticists in particular areas and other areas that are almost genetic deserts where there are very few providers. Even though this map is based on numbers of MDs and DOs, the same pattern holds true for our important colleagues in genetic counseling. So to put it plainly, we're not recruiting enough geneticists, and too few of us are from minoritized backgrounds. So what can we do? Well, one of the ways to explore this was to host these listening sessions. This was under the leadership of Ben Solomon at the NHGRI. And we hosted these listening sessions where 193 members of our profession came together to discuss the factors that influenced participation in the workforce. We saw lots of different things contributing to these traits or these outcomes. We saw both financial, scope of practice issues, but also exposure to mentors was a really important aspect that was lacking as well. Similarly, the ACMG was engaged in a workforce development and optimization study, and they also came up with a lot of the same kinds of factors that influenced recruitment, retention, and training. But if you look closely at this list, you'll see that many of these activities and points that were identified relate directly back to mentoring. So how can we increase our mentoring and increase exposure of our field to trainees? Well, one mechanism was through this virtual specialty forum that we started participating in a couple of years ago. Initially, when the forum was founded, genetics did not have a seat at the table, but through the advocacy of many, including Susan Klugman, we were able to now start participating in this forum and start actually making connections for these trainees. We found that it was really helpful, particularly for students who were in these kind of deserts where they didn't have access to mentors, to be able to make those connections one-on-one. And Mimi Blitzer was especially effective in making those connections for those students. Also, Michelle Takamoto and her work with the Alliance for Genomic Justice is another person who's helping to make important one-on-one connections for future geneticists in many different aspects of the workforce. Also, through my work at FACMG, we started to look at learners who were younger in age, and we started engaging in outreach activities. These outreach activities have been focused on Title I public schools, as well as career panels at medical schools. And I am here to tell you that the work in middle schools and high schools is incredibly life-affirming. You haven't lived until you've talked about de-extinction of woolly mammoths and a softball-sized woolly mammoth meatball with middle schoolers. Just trust me on that. And I promise we also talked about clinical genetics with them as well. So I really just want us to be thinking about this idea that genomic medicine is progressing at an incredibly rapid pace, and we have a great need for providers. So to enjoy the benefits of genomic medicine for all, it's going to take all of us. This is an all hands on deck sort of moment. If we want to be able to fully launch our mentees into this golden era of genomic medicine, we need to center this culture of authentic mentoring and authentic connection. That is what is going to help drive our mentees into this space and to develop a workforce of innovative, equitable, and diverse scientists in order to forward scientific discovery and health equity. I hope you'll join us on these important initiatives. Thank you.
Good morning, everybody, and it's amazing to be able to follow that great talk. And it's so nice to see so many friends and colleagues here at this meeting this week. So today, as my slides comes up, you'll hear that I'm talking about the village it takes to advance genomics medicine. And I think this is an incredibly exciting time to be a part of that village. So here are my disclosures not relevant to this talk. So, over the past crazy year, I've been thinking a lot about disruptive technologies in medicine, things that one day seem like science fiction and then the next become part of clinical care. One such example, liver transplant, pioneered by Dr. Thomas Starzl, shown here, sounds a little crazy on its surface. But I think as a field in genetics, we can learn so much from other medical subspecialties that have gone through these periods of rapid growth and change. Case in point, look at this picture. You see, the stoic, noble surgeon in the foreground. But what I love about it is the massive humanity behind him. No one person can do a liver transplant by themselves. We already know this in genetics. We know we have to collaborate and work together, but it's going to become even more important as new technologies are expanding. Another lesson we can learn is that sometimes the road is very rocky. Whether it's a liver transplant or new gene editing or gene therapies, these are high-risk interventions and things are going to go wrong at times. If you take a look at this timeline, the first 6 individuals who underwent liver transplant died in the peri-op period. It was so severe that the surgeons had a self-imposed moratorium on transplantation while they tried to figure it out. Eventually, by the late 60s, they figured out technically how to do this. However, a new challenge arose from this sick death. This is an interview with Thomas Starzl from the New York Times in 2009 that really struck a chord with me. They asked him about how his life experience changed his beliefs in healthcare. And if you look at this, he said, I came to realize that in transplantation, there was a kind of class filter through which ultimately, when transplant became profitable, that it was much easier to get a transplant if you had a lot of money than if you were poor. So he became a strong advocate for healthcare for everybody. We have experienced this as a field as we think about access to genetic services and genetic testing, and this difference is going to become even more evident when we're talking about high-cost medicines. However, despite all of that, it is an incredibly exciting time to be in the world of clinical genetics. A lot of people are starting to use this term interventional genetics, which I understand, but I kind of chuckle at some of the time. Because if you think about it, that's what we've been doing as a field for decades, right? Meeting a patient where they are, understanding their genetic diagnosis, and tailoring their care to that diagnosis. The only difference is now we're going to have a lot more tools in our toolbox. So to take a step back, how did I get interested in interventional genetics? Well, it started with a Friday afternoon consult when I was a young fellow, and I met this little guy and actually a couple of his family members. And as you can see, he has a very different phenotype than a typical newborn, very rigid, very hypertonic, and I had no idea what was going on. So it took a village. This is a village that came together to tackle this case. Some of the folks I think are in this room, but you can see we all came from very different backgrounds, different fields of genetics, but we worked together to figure out what was going on. And this was at the very early stages of clinical exome sequencing. And it was so exciting, and it's still exciting to think about the fact that you can meet a patient and figure out the molecular cause of their disease that has never been known to cause human disease before. In this case, we came up with a candidate gene. It made a lot of sense biologically. We don't have to get into all the details, but the idea is this kid had 2 genetic variants in a gene called ATAD1. What that meant biologically was that he had too many AMPA receptors on his postsynaptic neurons, which would lead to hyperexcitability and perhaps a phenotype like the one you saw in that video. But what was so exciting was at that time, just serendipitously, there was a clinical trial going on of a new drug for seizures that specifically blocked AMPA receptors. So knowing his genetic diagnosis allowed us to pick the right, at this point, experimental therapy for him. To make a very, very long story short, it had some benefit. It improved seizures, just showing you different parts of his EEG recordings, and it improved his tone, shown here. It wasn't a cure, but it did ameliorate a lot of his symptoms. And so I think using the information we had available to us by a transformational technology at that point, exome sequencing, really allowed us to improve the care of our patients. Fast forward, I guess, more than a decade at this point, and I want to tell a story about a very different team that came together, but with the same goal of trying to meet a patient where they are, understand their genetics, and pick the right tool to treat their disease at that point in time. This team is shown here, and there's a lot more folks that I'll talk about along the way, but I want to highlight this picture because of the person in the middle. That is now Dr. Sarah Grandinetti. For those of you who don't know, she was a graduate student that essentially led all of the work that I'm going to talk about. Essentially all the work to support a first-in-human trial of a personalized base editing therapy, which is pretty amazing. So for the trainees in the audience, I want you to know that your work really matters. Another member of this team is Dr. Kiran Musaniru, shown here in the gray sweatshirt. He is an adult cardiologist, right? He had never actually held a baby until we started working on this together. And so it takes a village of people with different expertise, different backgrounds to be able to come together to really push the frontier for our patients. And so we started working together several years ago now, thinking about ways to use new tools to improve genomic disease. The basic tool that I'm going to talk about a lot is this thing called CRISPR. It actually was not discovered when I started my clinical training, so things move quickly. And what you can see here is just a molecule that can derive from bacteria that can hone in to any part of the genome and evoke a change. It's directed to the genome by that purple molecule called a guide RNA. You can think of the guide RNA sort of like a GPS signal. Traditional CRISPR acts like a molecular scissors and just cuts the DNA. What we were trying to use was something called base editing, so a technology that was developed by David Liu that allows for specific transition variants to be corrected. You can think of it like a pencil in an eraser. We deliver this through a lipid nanoparticle, that blue blob that you're seeing right there. And so key takeaway of this that I want you to remember is all this is essentially reprogramming this pencil to go to a different place in the genome to correct a specific variant based on that guide RNA. So when we started thinking about how can you take this tool and apply it to genetic disease, we started brainstorming diseases where this might work. And what I'm showing you here is a lot of work in one slide, but the idea is we started with PKU, and you heard a beautiful talk about PKU earlier today. But the idea is that we came up with a way to develop a one-time base editing therapy for a new mouse model of PKU. Our PKU mice had very high phenylalanine levels, shown there in orange and green, about 2,000, similar to an untreated human. And I was amazed to see that you give a one-time dose, and within 48 hours, the phenylalanine levels entirely normalized in our mice. I didn't believe it. The graduate students all repeated it, and it happened over and over again. And the bottom is I have to show some mice in a talk, so you're seeing essentially the motor benefit of this type of therapy. But when we saw this, it really, for me, for the first time, opened up my eyes to the power of this new technology. But with that little bit of success, really, I started to worry about what would come next. Because we had chosen to go after one of the most common PKU variants, like a lot of industry folks before us, in all honesty, because we thought we would have enough patients to populate a clinical trial. But this type of technology requires you to know the diagnosis of your patients. But we in this room know there are huge swaths of the population where we don't know the full catalog of variants that lead to genetic disease. This is just showing you a map of where we have good catalog data for PKU. So what does this mean when we're talking about personalized therapy? Well, we have a huge potential to introduce bias into the system. If we, like others, focus on the most frequent variants and well-studied diseases with large commercial opportunities, it's going to be unfair. So what we decided to do was to turn this problem on its head and meet the patient where they are at the point in time of whatever their clinical diagnosis is, and figure out if the technology was appropriate for the patient in front of us, rather than a priori picking a genetic variant and working on a common variant. So we decided to really not care about how rare the disease was or how rare the variant, but just figuring out if the match of the tool and the disease made sense. So where did we start? We thought a lot about this, both the ethics of this and the practicalities of this, and we decided to work on urea cycle disorders. I'm a metabolic geneticist. I take care of patients with urea cycle disorders, and I've seen what these diseases can do to patients. But there's a lot of practical reasons why we chose this. The first is, and I know a lot of people in this audience have gotten phone calls asking this question, so hopefully this helps answer questions for patients. LNPs right now, so the tool to deliver those therapies, really only go to the liver. We cannot edit the brain with LNPs at this moment in time. So really we had to start with the disease where fixing the liver might have a benefit. In urea cycle disorders, we had biologic plausibility. We knew that fixing the liver would have a benefit because liver transplant works. The hard part is you have to wait till a child is big enough for liver transplant, and you can incur real neurologic damage or even death during that wait. The other reason we chose urea cycle disorders is there had been an amazing amount of work from many people in this room about the natural history of urea cycle disorders and the biomarkers. So we could essentially stand on the shoulders of giants to be able to leverage all of that data in our work. So how do you actually do this? And I know a lot of people have heard the story of our patient that we treated with a personalized base editing therapy, or experimental therapy, I should say, that had CPS1. Well, it wasn't just this timeline of 8 months or 11 months or whatever on the screen. We had been preparing for this and working in the lab and figuring out a way as a massive team that we could come up with an editing solution in real time to meet the needs of a patient in front of us. So then in 2004, in the summer, when our patient with CPS1 deficiency was born, we thought we might actually be able to try this for the first time. I'm not going to go through every step of this timeline, but what I will tell you are there's some key team members that made this possible. The first is our metabolic physicians and our ICU doctors who identified the fact that he had an elevated ammonia very early on and got him to dialysis as quickly as possible. A lot's been made about his neurologic outcome, but this is the reason why he's doing so well. Our genomic diagnostics lab and our metabolic labs were able to give us his answer very quickly within the first week of life. So then we were off to the races to do this crazy timeline of trying to figure out how you develop a drug, how you make a drug, how you make sure it's safe, and how you make sure that it's ethical to put into a patient for the first time. And a key member of the team here, every step along the way, was his family. They had no idea. We didn't really think that this would be an option for him, but they wanted us to advance the science to help others, and they were full teammates in this. So the village keeps growing with everything you try to do. So how did we actually do this clinically? I'm not going to go into the details a lot, but the idea was our goal was to maximize safety for him. With LNP editing drugs, you can give multiple doses, unlike AAV gene therapy. So he received 3 doses. I think our clinical experience made me realize more people that we need to bring into our village of clinical genetics. One is our immunology colleagues. We assumed he was CRIM negative. He had biallelic null variants. He didn't make any CPS1 protein, we think. And so we were worried about an immune response if he suddenly started making CPS1 protein. So we figured out an immunosuppression regimen. And then importantly, the decisions to redose were not made by me. They were made by a team of folks with diverse expertise that came together, including our medical ethicist and our bioethicist, to figure out what was best for him. Everyone asks what happened, and I think there have been conflicting stories in the media about what really happened, but here's my take on it. This is a single patient expanded access IND. So technically in our country, that's part of clinical care. I can't tell you definitively that this works. I can tell you what I observed, what we observed. Essentially, this is showing his timeline. The top shows his blood urea nitrogen level. The bottom shows his glutamine. And over time, with each dose, his biomarkers improved. Currently, he's doing really well. Initially, he was very protein restricted, but now he's able to tolerate a lot more protein. He still is on a low dose of nitrogen scavengers. We've been able to reduce it by about half, but we still treat him like he has a mild urea cycle disorder. He's off immunosuppression, and he's been able to tolerate a number of infections, including croup that required steroids without hyperammonemia. So what can we learn from this? Like I said, I really think we can say that his severe urea cycle disorder is a little bit milder, or is milder, I hope. And we saw that it's possible to do this if the stars align, that everyone drops everything and donates their time and resources, and this massive village comes together. That is not sustainable for scaling, and we need a better way to scale this. We need longer follow-up, but I will tell you that delivery to other organs still remains a challenge, and I'm happy to talk with physicians in the audience about that. So right now, this really only works, in my opinion, for liver diseases. I think we'll move other places sometime in the future, but we need to bring in more people, engineers, scientists, to figure out better delivery mechanisms. But ultimately, this situation introduces the possibility of great inequity and bias. We have to figure out how to scale this equitably. And the only way that we can do this is to think about this challenge as it comes up. So going back to this quote, right? This idea that we have to be advocates for healthcare for everybody. How does that work in our system? Well, in our system, there's discrepancies of who has access to clinical trials, who has the bandwidth to be able to have a parent that's pushing investigators to make a therapy for their child. So I feel the only way we can really get there is by scaling up formal clinical trials to get to approvals. Because approvals in this country is what allows things to be reimbursed. And I know reimbursement is a problem, guys. I know prior auths. I make those phone calls. But I will tell you, it's the only hope we have of getting this paid for more. So we've been working on this nonstop. This is just showing you some of our regulatory framework that we've been working with, funded through federally funded science. So again, thank you to the NIH and the Somatic Cell Genome Editing Consortium to figure out a way to scale clinical trials. We can't make a 10-year timeline with $10 million or whatever it takes for a traditional drug development for every rare disease genetic variant. So they've allowed us to come to a place where we hope to soon be able to launch a clinical trial that we're calling an umbrella of umbrella trials that can measure the safety and efficacy of these drugs across multiple urea cycle disorders, saving time and getting patients access more quickly. But phase 1-2 is just one step. The biggest hurdle is to get to that approval, and I know so many in this room feel that, especially right now, that approvals in the rare disease space are so important. So the FDA has announced this new plausible mechanism framework. The idea is that you might be able to streamline the approval process to essentially treat fewer patients for rationally designed drugs or rational pathway drugs. I will say we'll see what happens. The devil's in the details, but I do know one thing. If we're approving drugs after treating fewer patients, it is all of our responsibility to prioritize safety and transparency. We know data from every rare disease patient is precious, but even more so now. And it's hard, I think, as academics, as physicians, but we really need our industry partners to step up also and share what we are learning. Oh, I apologize. Okay. So last thing, how do we pay for this? We have to make the case to payers. In urea cycle disorders, we're able to do this because we know how much liver transplant costs. We know how much nitrogen scavengers cost. But we have to start calculating our worth clinically and the services we provide in order to make this case. But I think the biggest thing we need to do to scale and pay for this is to create the workforce. And yes, this is entirely AI generated, but I was really excited to see that when I asked ChatGPT to show me what an interventional geneticist looks like, they came up with a team that all looks like they might have different backgrounds or might be interested in different things. I have no idea what they're actually doing in this picture, but I will say, I think that it's an exciting time to be a young geneticist and even an older or aging geneticist. And I think we all need to learn these skills to work together because we have to have a seat at the table as this transformative technology comes into clinical practice because we know these diseases and we know our patients and we have to make sure it makes sense to put them into these new therapies or give them these new therapies. So with that, I want to thank the massive village that helped all of our work, including CHOP Metabolism and genetics and so many people from different backgrounds and our patients. So thanks a lot.
Thanks very much for the opportunity to be here with you today. And I get to clean up after these two wonderful speakers and bring together some of the threads that they brought to you, threads of mentoring and threads of the village, threads of bias, threads of collaboration. And what I'm excited by today is to share with you not only my story, but the story that I think is true for a lot of us and to bring some of this very much home. 31 years ago, this was not my field. I was a homeschooling mom with a background in theology, having been a college chaplain. And my two kids appeared for a couple of years, especially my daughter, with some bumps on the sides of her neck. I did the typical diagnostic odyssey at the time, not knowing at all that it was typical, and went from clinician to clinician, especially to my pediatrician, who was a friend of mine, saying there's something wrong here. She said there was something wrong and that it was in my mind, my brain, and that I really needed to go to therapy. So I went to therapy. Thank goodness, in fact. Continuing to search, though, that this just wasn't right. I looked and finally went out of, in those days, 1994, went out of plan, out of pocket a couple days before Christmas. And at that time, went to a dermatologist who happened to be boarded as an ophthalmologist as well, and began to learn about what systemic disease was, expecting that he was going to tell me, this is a laundry detergent problem. This is some sort of allergy. And instead what he said was, this is pseudoxanthoma elasticum. And my brother and my father had died the prior 2 years of different cancers. That oma at the end of pseudoxanthoma had me reeling right there in his office. He took a glance at my son and said, oh, look, he has it too. They're ages 5 and 7. And all of a sudden I'm in a world that I didn't really know at all. I went into some kind of massive neuroses to take action. My feeling was we can't have them experience what pseudoxanthoma elasticum, PXE for short, brings you. And that's vision loss to the point of being legally blind, claudication, vascular disease, and so forth. And certainly this skin, even the skin issue for me at that point was a lot to handle. I put together a research consortium. I brought together these great scientists. I put together a registry and a biobank, IRB, all sorts of things that were totally out of my wheelhouse, really, again, driven by neuroses more than anything else, that I did not want this to happen to my kids. And so I worked really hard to do all these kind of novel things in 1994, 95, when the internet itself really wasn't anywhere, including we had not yet finished the Human Genome Project. I had a lot of mentors along the way, including postdocs who at night at Harvard University gave me bench space between 10 p.m. and 2 a.m. and co-discovered the gene, not with them, but with some groups around the world. The gene is ABCC6, which happens to be expressed predominantly in the liver. And quite serendipitously, I'll be working, and I am working with Becca and Kieran and others now to see how we correct it, because it turns out to be a metabolic disease and not a dermatological and ophthalmologic disease, except in its phenotype, its expression. All of that brought me to a place where I was being asked over and over, could you do this for other diseases? And so I joined Genetic Alliance, which was then 10 years old already, founded by Victor McCusick in 1986, and brought the skills and the tools that I had created for PXE International, the little organization I created, to Genetic Alliance, and scaled it for as many diseases as we could, and have been there for the last almost 30 years. In each step of this process, my biggest drive was, how can I take the place of my kids? How can I take this away from them? How can I make this better for them? How can I change this for them? And all that meant that I did not enter this field of research and genomics and all the kinds of wonderful tools and technologies we see at a meeting like this for this reason. I came here to see if I could alleviate their suffering, if I could trade places with them. If I couldn't take their disease away, could I stand in their place? So I'm going to pause and give a minute of my talk to you in this form. I want you, for this next minute or so, which will seem like eternity, for you to imagine or remember, might be using your imagination, might be using your memory, of a time when you stood before someone else, probably a loved one, and said, I wish I could take this from you. I would trade places with you if I could take away what you're facing. So notice what arises in you when you notice that impulse, when you feel, I wish I could take that from you. I wish I could stand in your place. I'm of the mind that all of us here are here for that reason. Maybe we forgot that because of all the bureaucracy we deal with, the grant writing, the funding shifts, the deciding who gets funding if we're on the VC side. All the bureaucracy, all the red tape may have lead us to forgetting that that's why we're here, that we're in the business of alleviating suffering, that we're trying to figure out how can we step into somebody else's place, bring out something that would be of use to them. One person that I knew felt that as a great connect the dots for me at this meeting is Mira Irons, was the geneticist they sent us to. So the current president of the American College of Medical Genetics. I knew she knew we were sitting with a lot of uncertainty, a lot of fear, and that she wished she could take it away. And that meant a lot to me. We're here not just about sequencing and technologies and how cool AI is and how much it's going to bring to this field, but we're here because we want to make sense of uncertainty. Every single one of these talks has talked about the puzzle, the pieces we're trying to put together, the things we're trying to figure out. That uncertainty is something that we as parents and we as partners and we as children of loved ones deal with all the time. We humans don't like uncertainty. There are whole theories and methods and philosophies dedicated to our dealing with uncertainty. Buddhism says if we can just live with it, we would have no suffering. And we all know how hard that is to do. So our unraveling of uncertainty and our ability to be with and address fear is really critical. Genomic medicine ultimately offers a kind of hope. It's a certain flavor, certain way of being with this that's really important. Today, my children's disease still has no treatment despite my finding the gene and putting together several clinical trials and so forth. And I still have hope. And that hope is kind of what buoys us all up. There's many estimates about how many people in the world live with genetic diseases. 250 million, 300 million. You'll see all sorts of numbers on different slides. And in fact, I'm part of the Lancet Commission on Rare Diseases. Kim Boycott is leading that, and we're delving into that number. It's important. But in one way, it's not important. It's a lot of people. There are a lot of people who will benefit by and who are suffering with the things that we're addressing here. Most of these people are children, and most have never seen a medical geneticist or a genetic counselor. And that's not because the science is not here. We all know it is. You walk through the posters here, or walk through around the booths, listen to the talks. It's incredible what's happening. The biggest problem is that all of these technologies, just like any other technological field, remain concentrated in high-income regions. And it's people who are invisible to us who do not get access to these technologies. So we're changing that, and some of you here in the audience are part of that. Genetic Alliance has a program called iHope, and iHope is a program that was born at Illumina by my colleagues Ryan Taft and Aaron Venti, who are in the audience here today. Illumina gives us free reagents and philanthropic dollars for many years now to bring no cost whole genome, whole exome sequencing to children who live in low to middle income countries. It's been pretty phenomenal. Over these 12 years or so that the program has existed, about 3,000 kids have gotten free whole genome and whole exome sequencing. Our question really was, can we bring this kind of care, this kind of really advanced genomics care, to places like Zimbabwe and Ghana and Peru and Mexico and Vietnam? And the answer has been yes, yes, yes. This last year alone, we did more than 1,000 kids, trios, and brought them some answers. Sometimes we don't bring them answers. So about 50% receive a diagnosis, but about 70%, regardless of whether they got a diagnosis or not, are seeing a change in management for all the reasons you could imagine, that we're able to do less testing that's not needed or imaging, less interventional things that aren't relevant, bringing ourselves to a place where ultimately, I believe, we are sitting with each of those kids and their clinicians, because back to it takes a village, and their advocates and their psychologists and their whole team to be with them as they face this disease, whatever it is. And so this program has benefited certainly from the incredible donation of Illumina all these years, from now PacBio joining us, and then also pro bono labs. We have 6 of them around the world. Some of you are here in the audience. We met the iHope team on Monday, the network, and about 25 clinical sites. Again, some of you are here in the audience who are referring these children through us to these labs. And so this system's working extremely well, and it's bringing hope to these children. In this process, we have also integrated a piece that was really important to me from the very beginning, which is that people control their own data, genomic data, clinical data, through an incredible technology, and I'll say that as a proud mom, called Digital Cabinet, something my son created over all these years. He will tell you from when he was 5 and looking in the file drawers in our basement to now when he's running an international data system that's using large language models, AI, and other things to bring all these data together. And ultimately, the most important piece, to allow people to connect to their own data and decide how it will be used, so that beyond this either diagnosis or no diagnosis, people can go and do all the things that I had to do a lot of heavy lifting for all those years ago, and be able to find someone else who understands how it is to live with this disease, to find other people who are interested in moving the research forward, to find clinical trials, and certainly to find support. Genetic Alliance over our 40 years has put together more than 10,000 support organizations under our umbrella, and now we get to deliver all of that to these individuals who are greatly in need. A key insight of this was, and this is not going to be terribly popular here, but we don't need elite centers and experts to be able to do this work. We've done it in places where pediatricians, genetic counselors, advocates like myself in countries in low-income countries are the ones connecting the dots and bringing the children to our program and delivering back the results. We have a poster 505, I think it is, and you can go and see this technically laid out. I'm going to only speak to it on a high level. But the idea that, again, the village can be involved, the village can bring these kinds of answers to people, this kind of support, this being with. The human impact is enormous of what we're doing because people feel cared for, they feel seen, they feel heard. Every single one of us who goes through the diagnostic odyssey believes we are alone, and we're not, because everybody else is going through the same one. But in that terror, in that fear, in that aloneness, we're magnifying, in effect, the ramifications of the disease because we layer on it all these human emotions that we all deal with. From this work, we've learned and we've actualized 3 things. First, deploying the genomic technology is not the hard part. The technology exists, again, as we've all seen. And what's really scarce, though, is the willingness to restructure power and pay attention to those who are invisible to us. The second thing is patients are not subjects. This has been a drum of mine I have beat for a long time. I believe calling us subjects really diminishes who we are and what we have to offer. We're participants. We're here. We're part of this. And so as people begin to own their own data, and you all can have heard all the arguments against this and all the issues around, oh, this will silo it and things won't happen the way they need to and so on. What we've in fact found, and I've found this over my 30 plus years of working in this, is when families govern what happens, they don't destabilize science. They in fact accelerate science. And I think we're seeing that more and more. And the third thing is, the next frontier is not more complex genomics and technologies and so on. Certainly, we're going to continue that, and we'll see that year to year here. Even as AI itself continues to improve the tools that we're talking about and itself, I think what we're going to see is the next frontier is equitable deployment. Some of you here, a few of you at least, are as old as I am, and remember that we sat in this meeting and many others when the human genome was being completed, and what did we hear? We heard from Francis Collins on down, this technology will not create greater inequities. I don't know. I think we've created some more inequities, and I know that iHope is trying to break down those inequities, to break down those silos, and I know and hear from many of you that you're concerned with that too. So I think we really need to pay attention to this piece that's so critical about this being the real frontier. Today, we might continue to capitulate on this principle that we should have equity because we're facing some very, very great pressures that we can't ignore outside this room, that we wake up in the morning and hear again and again. And some of the ones that boil down for us very clearly are erratic funding, global inequities overall, and political unrest, and a kind of xenophobic contraction. Every one of these contractions, and every time we contract, I think we see more clearly what is unethical, what is not right, what is unjust. And so as much as we hate what's happening in this regard, for us personally, for those around the world, I think it brings into focus a North Star for us if we pay attention to that. I think this is the moment where we decide, are genomic technologies, genomic medicine, genomic care going to be a luxury, or are they going to be for the public good? I think that is a very big question before us. In my TED Talk more than 10 years ago, I said, we don't want to be invited to the table. We don't want a seat at the table. We want to create the table. We, the people, the participants, the advocates, the moms and dads. We want to decide what's the menu. People said to us, if you're not deciding the menu, you're on the menu. We don't want to be on the menu. We want to decide it. We want to decide who are the guests that come to the table. So a lot of forward-looking, let's make a difference here. Let's take the bull by the horns here. And I'm going to add a piece to that today, 10 years later, and I'm going to say the system must also move toward the people who suffer, the people who are invisible. We cannot count on those who are already burdened, already suffering, to do the work. As much as I'm glad that I've been able to build systems and I've been able to mentor hundreds of advocates to do the same thing, I'm also really aware, and probably primarily by my son, who's now 37 years old and has a 5-year-old or 8-year-old, by him saying, we are not interested in building another advocacy group. We're not interested in a new registry. We don't care about a biobank or a clinical trial. We want to sit on the couch and watch Bluey with our kids. That's our goal. And that just struck me like, whew, I have forgotten to some degree what we're here for. And so how is it that whatever your equivalent is, we bring people to a place where they can be with their kids in the way that he's talking about? So this system that we're building here must move toward those who suffer. 31 years ago, I wanted to trade places with my kids. I still want to trade places with my kids. I do not want my kids to be faced with what it means to be legally blind, to be unable to be healthy and vigorous, to have cardiovascular disease, all those things. If you think back to who you imagined or what you imagined at the very beginning of my talk here, and imagine again that person or persons, I bet you still feel, even if it was years ago, that you would like to see that pain go away. So we can't trade places biologically. We can't trade places structurally. But we can trade places in such a way that we start to bring what it is we have to the people we love. Some people worry about losing their jobs to technology or artificial intelligence. But I think the job in front of us is clearer than ever, and artificial anything can't solve it. The moment that we pause and turn towards someone else, even if it's the person as you're leaving the auditorium today that's beside you, that's near you, and are present for them, even for a moment, I think we start to offer who we fundamentally are and what we really have to offer. Yes, all the wonderful stuff that comes out of our brains and builds all this cool technology, but also our humanity and who we are. We offer something more than analysis. We offer company. We offer hope. And I think this is how genomic medicine and the resulting care becomes a public good. And this is how we stay rooted in what matters. Thank you.